So what's remarkable, there's some bats, like the horseshoe bats, that carry these SARS viruses around with them in their body, in their bloodstream, and nothing happens. They just carry it with them. They don't even get sick. And so we'd like to know, why is that bat species and a few others resistant to this virus, and why some humans are also getting infected, match for age, match for sex, match for pre-existing health conditions. Some other person will get seriously ill and maybe even die. And so we're trying to see if those humans and those bats share something similar in their immunity. My everyday job is trying to understand the genetic and neural mechanisms of language. I got involved in generating high-quality genome assemblies, putting genetic material together. If I want to identify what's similar between humans and parrots that chimpanzees don't have, I need high-quality genomic data. And so when this COVID pandemic happened, I thought we can take the same techniques that the Vertebrate Genomes Project developed to apply it to generating high-quality genomes of people and of different species, to put together their immunity genes to try to understand the vulnerability and resistance that different species in humans have to this virus. We're going to look at the ACE2 receptor initially. It's the receptor that the virus binds to that allows it to get inside the cell and start to replicate. But the ACE2 receptor is not where things stop, because like the horseshoe bats, the Chinese horseshoe bat and the pangolin who get infected by these viruses have the ACE2 receptor. The virus goes in, but they don't get sick. So there's something else going on in their genome that's preventing them from getting sick. And this is why we want to sequence not just the ACE2 receptor across these species in different humans. We want to get the entire genome. So we have all the genetic material completely assembled without errors, and then scan the entire genome, including their immune genes, to see if once the virus gets into a person and they become a super spreader but don't get sick, what is preventing them from getting sick? And by having their genome, we'll be able to figure that out. So once we find these genetic differences between different species and between different humans within our own species that allows somebody to be resistant to the virus, we can take that genetic mutation and then target specific drugs against it, or actually synthesize gene products like antibodies to try to prevent infection. When I think about whether the research we do now will help resolve something for the current pandemic, I don't know the answer to that. And so what I hope can at least come from our current research is that it will help the next pandemic, whether that be in 100 years, 50 years, 10 years, or 200 years from now. We will learn something from the current project that will help us be ready for the next time around, because it's going to happen again.